genetic association studies of high-quality traits, and hopefully, the slide will stay on. Before going into the science, I really wanted to thank the organizers. This has been really great. I've been at CGSI last year. This is the first time at the Big Bear retreat. It's been a wonderful experience, and I very much look forward to this first short program. I'm going to be around till end of next week, so I'm more open to this for interactions. Great, so I'm going to start with a motivating slide. Why we do genetic association studies. We've heard this topic a bit in the last few days. I contended DNA genetics is a bit of a special layer because we get genetic information at conception, and then this information is constant across all the, approximately constant across all the cells in our body and over time as we age. And so if we find an association between a genetic variant and the phenotype of interest, or for example a disease, this points us towards a gene or a pathway that is upstream the manifestation of disease. And so we can use this as an initial clue to try to understand the pathways and the processes that are implicated in the manifestation of the disease. And we've known this for quite some time. We've been doing genome-wide association studies for now 20 years. And I know this has been a topic these days, but I'm going to recap very briefly the data type of analysis in GWAS, because that's a very important topic for this talk. So what we have typically, we have a phenotype of interest. This could be BMI, that we measure in a population of up to hundreds of thousands of individuals, maybe also millions of individuals. And then in the same set of people, we also have genetic variation data. And GWAS typically focus on common variants. We have tens of millions of them, and most typically on the most focus is on single nucleotide polymorphisms, SNPs. And the computational analysis is relatively simple. We use simple models like linear models with Gaussian noise, where we test for association between the phenotype and each single genetic variation in isolation. And from this test, we get a p-value that we use as a metric to establish the strength of the association between the value and the phenotype. And a typical representation of the output of this analysis is a Manhattan plot. Here, each circle is a variant. On the x-axis, we have the position of the variant, so you see different chromosomes. On the y-axis, we have the strength of the association between the variant and the phenotype. And this is quantified by minus log 10 p-value of the association test. And since we do many tests, of course, we need to be very strict when accounting for multiple hypothesis testing. So we have this genome-wide significant threshold of 5 × 10⁻⁸ that corresponds to family-wise error rate 5% approximately. And for a very long time doing GWAS, we were not finding many significant loss of any genes. Things have changed with increasing sample sizes, and when we get to the tens of thousands, then to the hundreds of thousands, now we find many genes. So this is an example from a GWAS of Alzheimer's disease that was published 5 years ago in Nature Genetics. And as you can see, we find tens of variants with complex traits. But now the challenge is how we actually go from this list of genes that we get, list of loss that we get, to actual disease biology. And so I asked ChatGPT and DALL-E to generate an image that sort of conveys the sort of sense of frustration of Gräf scientists as basic geneticists, that they have this list of genes, but then they still wonder, okay, but now what's the functional mechanism? How do we get from this list to actual disease biology? And I think this was a beautiful talk this morning, telling us how you can do that using gene expression and single cell data. And one of the first questions that one can ask, what's the cell type, what's the tissue implicated, what's the biological process, and so forth. So nowadays we're lucky because we many times measure a lot of different intermediate phenotypes in the same population cohort. These traits can span from single cell RNA-seq or cell level traits to medical imaging, MRI, histopathology images, and also clinical readouts such as blood test values, metabolites, psychological tests. And what we do in the lab, we develop machine learning methods that enable the genetic analysis of these high content phenotypic modalities, really to try and understand which are the intermediate traits implicated in the establishment and progression of human disease. And we apply this concept across many different data modalities, but today I'm going to tell you part of this focusing on genetic analysis of histological traits. This is a project that's been led by Cham, PhD student in the lab. Okay, so very briefly about the histological slides. H&E stain holds red images. H&E stands for hematoxylin and eosin stained holds red images. These are two dyes that emit in the visible that track cell nuclei, cytoplasm, extracellular matrix, and enable the visualization of cellular and tissue structure phenotypes. The microscopy images and they're used routinely in the clinic for disease diagnosis. They're very much used for cancer, but also other non-cancer diseases like steatohepatitis, chronic liver inflammation. As mentioned, they showcase detailed cell and tissue phenotypes that maybe you can study genetically to find disease mechanisms. So this is an example from a slide from GTEx, side of the liver, and these images they contain a lot of information. They take this space up to 4 gigabytes. They consist of millions of pixels. So if we zoom in, we can start seeing some of interesting tissue structure, but we can zoom even more and start looking into very specific cell-level phenotypes. Like here, we see a specific form of apoptotic death of hepatocytes that known as hepatocyte ballooning. This happens in chronic liver inflammation, and that's another example. But we zoom in another region, we see the presence of connective tissue. This is also known as fibrosis. This is the process that leads to dysfunctional liver, through cirrhosis, and typically also liver transplant. So we can measure all these traits, but so far there's no method that automates the quantification of these phenotypes in an unbiased way and enables genetic analysis. That's what I'll be talking about today. Our approach that we introduced, we call it HistoGWAS, GWAS of histological data, and it consists of 3 main components. First, we learn a semantic autoencoder of tissue patches. So we learn an encoder that projects these images of tissue into a latent space. We refer to it also as an embedding space. And then in this low-dimensional embedding space, we can do a bunch of statistical analysis, such as genome-wide association studies. And then we also train a decoder that enables us to go back to the high-content imaging space. In the latent space, then we do genetic analysis of these embeddings, and this is very similar to the models I was showing you before, but now instead of testing for association between a single variant and a single phenotype, we test for association between a single variant and the whole embedding. And finally, if we find genome-wide significant variants, we can use the decoder to visualize the genetic effects of the images using principles that are very similar to what you've seen in standard machine learning papers where people can interpolate between phases, put the glasses on, glasses off, and do other types of transformations. So I'm going to talk about these 3 in detail. Before a bit about the data, you heard about this before. It's genotype tissue expression dataset, GTEx. This consists of data from 54 tissues across about 1,000 individuals, and from which we have expression, histology, and also genetic information. And in this analysis, we focused on 11 tissues for which we had the highest sample size, more than 750 individuals. Great. So I'll start from the semantic autoencoder and how we train it. So I'll start telling you about how standard autoencoders work. So typically, for this model, you train both the encoder and the decoder jointly. The encoder goes to this compressed representation, and the decoder goes back to reconstruct the input. And typically here, you optimize a reconstruction loss. This could be a NEL2 loss, as you see here, between the reconstructed image and the original image. And this is directly operates on the intensities on the pixel level values, so the RGB values. So when you train several models, of course, compressed representation, since you try to reconstruct the whole image, tries to preserve all the information that you see, not only the semantic information. If we want to do a bit better, we think we can lose all the information, we just want to preserve the semantic concept. So to do that, we explore this strategy where we train the encoder and the decoder separately. So we first train the encoder. This is an example. We can do it using contrastive learning. So given two images, we can transform them through augmentations. These are transformations that we believe do not change the semantic content of the image. So for example, rotations, flips, color jittering, bit of zooming. And then what we do, we train a convolutional neural network that extracts embeddings, such that embeddings of different augmentations of the same image are similar, while embeddings of different images are different. And in this way, you learn to extract embeddings that are invariant to transformations that preserve content. And once we learn this encoder, we then try to revert it, training a generative adversarial network. So we start from a real image, of course, through the encoder now we can get the embedding. And then we have two agents here. We have a generator that takes as input the embedding, the real embedding, and the Gaussian noise, and generates a fake image. And then we have a discriminator that takes as input a pair, an image embedding pair, and needs to discriminate which pair is fake versus real. So now by training these two agents against each other, the generator gets better and better at generating fake images conditional on the embeddings, and the discriminator learns to distinguish real pairs from fake pairs. So the nice thing of this all happens conditionally on the embeddings that we learn from the encoder. So we learn to invert the process, the encoding process. And of course, we're going to use the generator as a decoder. So we apply this method across 11 tissues and analyzing about 20 million patches. Each one of them is a 256 by 256 image, and bringing across this data just to give some intuition about the scale of the problem. Great, so now we can study it all together. So start from real data, we encode them into an embedding, and then through the generator and Gaussian noise, we can go back and get semantic reconstructions. And as you can see here, we do this auto-encoding strategy is not attempt to reconstruct the whole image pixel by pixel exactly, but it wants to preserve the information, only the content information, so that is invariant to these augmentations that we have in the encoding procedure. And we can also sample different realizations of the same content. Here you can decompose this content axis versus realization of the content. And you can qualitatively notice some how what the embedding is capturing versus what's in the noise. Okay, but how do we quantitatively assess now that this auto-encoding is doing what we want to do? So we use gene expression for doing that, and we select an encoder that can best predict expression. And to do that, we evaluate different histological foundation models. We heard about foundation models a bit this morning. These are large unsupervised models that have been pre-trained on a very vast amount of histological data. And so we get patch level embeddings from these multiple encoding models. Then we obtain sled embeddings, just simply taking the mean of patch embeddings within a single slide. And then for each gene, we train a linear model to predict its expression from this slide level embedding, train on 50%, evaluating the remaining 50%, and then we assess association between the predictions and the observed expression values out of sample. And we compare these different models. This is a QQ plot showing the observed minus log 10p versus the expected, and each color here is a different model. And as you can see, the contrastive approaches perform best across the ones that we consider. And the standard encoder actually, since tries to reconstruct the whole image, it's not very semantic, is the one that performs worse across the models that we compare. But how do we prove that this semantic auto-encoding procedure actually preserves information we care about? And to do that, we can given the reconstructed patches, we can reuse the encoder to get embeddings of the reconstructions, right? And then we can say, okay, now if we apply the model that we used before to predict expression, but giving as input these the embeddings of the reconstructed patches, can we predict expression? Do we preserve our capabilities of predicting expression after these autoencoded patches? And yeah, when doing that, we can see this is the signal that you get from the observed data. And then if you reconstruct the semantic autoencoder, you preserve a lot of it, and you lose a lot of information using a standard autoencoder. Here again, each point is a change to analogous point with what I showed before. Okay, now I'll get to the second part. So we have now this autoencoding framework. Now we want to do genetic analysis in the latent space, in this embedding space. First of all, there's a lot of heterogeneity in these HNE-stained whole cell images. And so one way we address this, we perform an unsupervised analysis to identify a cluster of similarly looking patches. And then we perform our genetic analysis at the slide level within each cluster. We also assessed that these clusters also are associated with different parents of expression. So how we do GWAS in practice? So we use a generalized linear mixed model with a binomial likelihood. Here the genetic variant is the output, and then the likelihood is a binomial with two trials, and we model the logit of the rate using a linear model where we have a contribution from covariates. Here we account for genetic principal components, for population structure, and then we have an effect from these cluster signature embeddings. We model this as a random effect, and ultimately we assess the non-zero effect of these cluster signature embeddings through a variance component test. Some of you may know a sequence kernel association test, SCAT. It's very similar to this procedure where we assess that the variance of these weights is greater than zero. It's a bit of a technical slide, but from more intuitive level, what we do, we use this model to assess association between the embeddings and the single variant, and then we apply this test for each variant transformation for all the variants that we have access to. Okay, so these are the results that we get across 68 cluster signatures across 11 tissue. So we find 4 genome-wide significant variants that pass fairly-wise over 20%. We compute this threshold with permutation procedure. And now I'm going to go through some of the findings that we have and how we interpret them, how we use the decoder to interpret them. And I actually should say, I learned a couple of days ago that I should say how we explain them, not interpret them. The top variant is a variant on chromosome 9 that we find associated with the histology changes in thyroid. And this is an intron variant in the papillary thyroid cancer susceptibility candidate 2 gene, PTCSC2. The locus has been previously implicated with hypothyroidism in the GWAS infringement, and when we check association between the lead variant and gene expression, we see association with 128 genes that also reach for pathways implicated in hypothyroidism and thyroid cancer. So here you can see a locus zoom plot where we basically are zooming in the Manhattan plot we were looking at before in the smaller region than 1 megabase. And this is the results from our GWAS in thyroid. And here these are the results from the FinGen GWAS of hypothyroidism. So we can see a very strong localization between the two signals. And this variant is false within the PTSC2 gene. But now how do we get to interpret what's actually changing between the two? So for that we use the decoder. So the idea is that through the linear model that I was showing you before, we estimate the direction of effect in this embedding space. So once we know the direction, we can actually linearly interpolate in the embedding space and then project this linear interpolation and linear interpolated embeddings back to the image space and visualize a candidate phenotype that is being affected. And so we show this to 2 pathologists, and we found that this G allele is associated with a large follicles and increased color, these two structures in the thyroid are responsible for hormone production. So this is most likely a compensation mechanism. So when your hormone production lower than they should be, these structures tend to enlarge to compensate. And that's very well aligned with the Goiter phenotype and the type of pathogenesis that occurs in hypothyroidism, which is the disease for which we have the GWAS here. We also have another notable example in esophagus mucosa, where we find this variant is an intron variant in interferon-induced protein 44 gene, EF44. This is a grammatical chronic inflammation, and again, if you look at the top variant, these are associated with changes in expression of 30 genes. And yeah, we're interpreting this, we're showing these two pathologies. We could quantify that LIB-A was associated with notable enlargement of epithelial cell. Great, yeah, and then you can do also fancy movie. So, the nice thing of it is that you, since you have this stochastic decoder, you can also get different realizations of these effects, and so this could help, a pathologist to understand what may be driven by noise or what's robust type of different realizations. Of course, I think I would contend we always need a pathologist to make sure. It's a tool that is supposed to help pathologists in interpreting these associations rather than replacing them. Great, and then we also perform the power analysis to see how whether this method scales and how power increases with sample size. And so we simulated different cohorts, increasing sample size and varying the variance explained by the genetic variant of these embedding phenotypes. And in our analysis, we analyzed a relatively small cohort, right? This was in the range 650 to 1,000 individuals. And here we have high power up to variance explained 0.2%. But if we go to 10,000, then we can mark with very high power, very small effects, so up to 0.02%. So these results really show that this method scales, and this is possible. And with larger cohorts, this can unlock quite a lot of insights in terms of disease mechanism. Yeah, with that, I'd like to conclude. So I introduced HistoGWAS. It's a new framework integrating AI and statistical models for automated genetic analysis of histological traits. The automated part comes from this idea, using these large unsupervised models to automatically phenotype and then do discovery in an unbiased way, really optimized for scalability. Also under what we learned in the last few years, the main driver of discovery is sample size. So even if you're a bit imprecise about this phenotypic quantification, if this can be done, then we can just push sample size and discover more. Then I showed first ever automated GWAS for histology. It actually finds, what significant hits, 2.4 variants with some notable examples. And of course, the thin power simulations showcase that this method can actually be used, and we can learn a lot from these findings when increasing sample size. And so hopefully this will open a new direction, new type of genetic analysis in the next few years. Okay, with that, we have a preprint out, so if you're interested in some technical details, please check it out, or just talk to me too. I want to thank my group and collaborator, Campus Munich, funding, and specifically people highlighted in pink, Shwamborn in particular, the student who was driving this project. And with that, thank you for your attention. I look forward to questions.